The overall goal of this procedure is to be able to quantitatively measure cellular motility over time using a gold nanoparticle-based cell motility assay. This is accomplished by first preparing glass cover slips for a coating of gold nanoparticles. Specifically, endotoxin-free cover slips are coated with gelatin and then baked. The second step is to reduce chloroauric acid, which will create gold nanoparticles, and to coat the gelatin-coated cover slips evenly with the solution. Next, the cells are placed onto the gold nanoparticle-coated cover slips for the desired time frame of the experiment. Cells moving on the gelatin will displace gold nanoparticles, creating tracks. The final step is the monitoring and quantitation of cell motility using microscopy to image the tracks created by the moving cells. Ultimately, area measurements of the tracks are performed using freely available software.
One of the main advantages of this technique over existing methods, such as time-lapse imaging, is that this assay requires only a standard light microscope, standard camera, and uses freely available imaging software. In addition, the assay also easily allows the analysis and comparison of multiple samples using a method that is capable of high throughput screening. Although this method can provide insight into the influence of different physiological factors on cell movement, it can also be applied to other systems, such as the studies on the effects pathogens have on the motility of infected cells.
Generally, individuals new to this method may struggle because the method requires one to strictly follow volumes and temperatures indicated in the protocol. In this protocol, an emphasis is placed on assessing the color of the final